glucose can be broken down to 3-carbon pyruvate through glycolysis. On the other hand, pyruvate can be used to synthesize glucose through a process called gluconeogenesis. Glucose can also be converted to glycogen for energy storage through a process called glycogenesis. When we need ATP, glycogen can be degraded to release glucose. This process is called glycogenolysis. Further, glucose can be converted to ribose 5-phosphate for nucleotide synthesis through a process called HMP Shunt. In this lecture, we will focus on these 4 processes. We will cover the following 5 topics. We will start with glycogen. First, let's look at glycogenesis. Glycogen is the storage form of glucose. Glycogen synthesis and degradation occurs primarily in liver and skeletal muscle, although other tissues, including cardiac muscle and the kidney, store smaller quantities. Glycogen stored in the liver is a source of glucose for the whole body. Muscle glycogen is stored as an energy reserve for muscle contraction. In fast-twitch muscle, glucose is converted primarily to lactate. In slow-twitch muscle, glucose is completely oxidized to carbon dioxide. Here is a glycogen complex. Every glycogen granule has at the center a glycogenic protein. To initiate glycogen synthesis, a primer is required. The protein glycogenin serves as the primer. Glycogenin is an enzyme that attaches glucose from UDP glucose to tyrosine residues. Here is UDP glucose. This glycosylated protein then serves as a primer for glycogen synthase, a rate-limiting enzyme for glycogen synthesis. UDP glucose is synthesized from glucose 1-phosphate and UTP. Glucose 1-phosphate is derived from glucose 6-phosphate from glycolysis. Glucose from UDP glucose will be transferred to a glowing glycogen. This picture summarizes glycogen synthesis. It starts with glycogenin. This is glycogenin. It adds the first glucose to its tyrosine residue and continues to add 7 more. Now, glycogenin is primed. Glycogen synthase comes in and adds more glucose to the non-reducing end. Branching enzymes comes to create branches. This picture shows you in a glowing glycogen. Glucose residues are joined together through alpha-1,4 glycosidic bond. Here is the non-reducing end. And here is a new glucose coming in the form of UDP glucose. Glycogen synthase adds the new glucose to the non-reducing end of glycogen. To keep glycogen packed in high density inside the cell, glycogen requires branching. This is done by branching enzyme. Here is a growing branch of glycogen. Branching enzyme transfer about 7 glucosyl residues from the non-reducing end of a branch to a new branch, at least 4 residues away. Here is an example. 7 glucosyl residues are removed from the non-reducing end and transferred to a new branch point. And this is an example of glycogen branching. Here is clinical correlation. Deficiency of branching enzyme causes Anderson's disease. Glycogen from patients remains very long unbranched. Now let's move to part 2. The rate-limiting enzyme of glycogenolysis is glycogen phosphorylase. This enzyme breaks alpha-1,4 glycosidic bond using inorganic phosphate. The product is glucose 1-phosphate. Glycogen phosphorylase requires pyridoxal phosphate, also called vitamin B6, as a coenzyme. Glycogen phosphorylase degrades the glycogen chain at the non-reducing end until 4 glucosyl residues remain before the branch point. This structure is called limit dextrin and needs another enzyme to remove it. That enzyme is debranching enzyme. Debranching enzyme removes 3 glucosyl residues from the limit dextrin and transfer to the next branch, and cleave the last 1 glucosyl residue and release it as a free glucose. Debranching enzyme has 2 activities. First is glucosyl 4,4 transferase that removes outer 3 glucosyl residues and transfer them to the non-reducing end of another chain. Second activity is amylo alpha-1,6 glucosidase that breaks alpha-1,6 bonds and release a free glucose. Here is clinical correlation. Cori's disease is caused by debranching enzyme deficiency. The enzyme deficiency leads to accumulation of limit dextrin. Patients have hepatomegaly. To help you remember Anderson's disease and Cori's disease, here I use one mnemonic: A, B, C, D. AB refers to Anderson's disease caused by branching enzyme deficiency. CD refers to Cori's disease caused by debranching enzyme deficiency. These slides show you 7 types of glycogen storage disease. Among them, type 1 to type 5 are high yield. Type 3, 4, and 7 have been described to you earlier. Von Glerke's disease is caused by glucose 6-phosphatase deficiency. The deficiency leads to trapping of phosphate in glucose 6-phosphate. Hepatomegaly and hyperuricemia are reminiscent of hereditary fructose intolerance. In addition, von Glerke's disease has lactic acidosis. Pompe's disease is caused by lysosomal alpha-1,4 glucosidase deficiency. It features muscle weakness, hepatomegaly, cardiomegaly, and heart failure. Myocardial disease is caused by muscle glycogen phosphorylase deficiency. It features exercise-induced cramps, rhabdomyolysis, renal failure with myoglobin urea. Patients have second wind phenomena. Here, 3 diseases that have hepatomegaly are summarized in one mnemonic: GDP. G refers to von Glerke's disease. D refers to debranching enzyme deficiency. P refers to Pompe's disease. Now let's move to part 3. First, glycogen synthase and glycogen phosphorylase are hormonally regulated to meet the needs of the body as a whole. Second, the pathways of glycogen synthesis and degradation are allosterically controlled to meet the needs of a particular tissue. In both cases, phosphorylation serves as a switch. For example, when phosphorylated, glycogen synthase is inactive. while glycogen phosphorylase is active. Vice versa, without phosphorylation, glycogen synthase is active and glycogen phosphorylase is inactive. Phosphorylation of glycogen synthase and glycogen phosphorylase is controlled by protein kinase A. Dephosphorylation is controlled by protein phosphatase 1. Then, what controls protein kinase A and protein phosphatase 1? Two signaling pathways provide an answer. Glucagon signaling activates cyclic AMP-dependent protein kinase A. Here's protein kinase A. And protein kinase A phosphorylates both glycogen synthase and glycogen phosphorylase. Glycogen synthesis is inhibited. Conversely, during fat state, insulin signaling activates protein phosphatase 1. Both glycogen synthase and glycogen phosphorylase are dephosphorylated. Glycogen synthesis increase. Allosteric regulation allows a rapid response to the needs of tissues, such as muscle and liver, and can override the effects of hormone-mediated covalent regulation. Glycogen synthase has one effector, while glycogen phosphorylase has 5. They are shown in one table. These effectors can be summarized in one mnemonic: 1, 2, 3. 1 refers to the fact that glycogen synthase has one positive effector, which is glucose 6-phosphate. 2 refers to the fact that glycogen phosphorylase has 2 positive effectors, calcium and AMP. 3 refers to 3 negative effectors for glycogen phosphorylase, glucose 6-phosphate, ATP, and glucose. In the muscle, calcium and AMP can override the control by insulin. Calcium and Calmodulin form a complex that activates glycogen phosphorylase kinase. Here is glycogen phosphorylase kinase. This enzyme can be activated by calcium and Calmodulin complex without phosphorylation. Upon activation, this enzyme can phosphorylate glycogen phosphorylase and make it active. AMP allosterically activates glycogen phosphorylase. Here is glycogen phosphorylase. AMP can directly activate this enzyme without phosphorylation. Calmodulin calcium complex is an essential component of many calcium-dependent enzymes. Besides control of glycogen degradation in muscle, Calmodulin calcium complex also control glycogen degradation in the liver. For example, during stress, epinephrine induces calcium release from ER. Calmodulin calcium complex activates glycogen phosphorylase kinase without phosphorylation, which activates glycogen phosphorylase. At the same time, Calmodulin calcium complex activates protein kinase C. Protein kinase C phosphorylates glycogen synthase, making it inactive. Now let's move to part 4. In fasting, glycogen reserve drops dramatically in the first 12 hours. During this time, gluconeogenesis increases. After 24 hours, gluconeogenesis represents the sole source of glucose. Gluconeogenesis occurs in the liver and the kidney. Important substrates for gluconeogenesis include glycerol 3-phosphate, derived from degradation of triacylglycerol. Lactate, from Cori Cycle. Glucogenic amino acids, from degradation of proteins and alanine cycle. Here is Cori Cycle. This is the liver, and this is muscle. During fasting, lactate from exercise in skeletal muscle or red blood cells is converted in the liver to glucose that can be returned to the red blood cells or muscle. This picture shows you Cori cycle occurring between the liver and the red blood cells. Here is alanine cycle. Muscles release alanine, delivering both a glucogenic substrate, pyruvate, and an amino group for urea synthesis. The liver converts pyruvate to glucose that can be returned to the muscle. Glucogenic amino acids can be used to synthesize glucose through gluconeogenesis. Ketogenic amino acids can be converted to acetyl-CoA or acetyl-acetyl-CoA. This table shows you except leucine and lysine that are pure ketogenic. All other amino acids are glucogenic. Here is the overview of gluconeogenesis. On the right side of this picture, from top to bottom, represent glycolytic pathways. Left side of this picture, from bottom up, represent gluconeogenic pathway. As we know, 3 steps in glycolysis. Specifically, these 3 steps are irreversible. These 3 steps are replaced by 4 steps unique to gluconeogenesis. 4 steps. These steps specifically include pyruvate carboxylase in mitochondria, PEP-CK in the cytoplasm, fructose 1,6-bisphosphatase in the cytoplasm, glucose 6-phosphatase in the lumen of ER. The first bypass from glycolysis is to convert pyruvate to PEP. This involves several steps. 2 key steps are catalyzed by pyruvate carboxylase and PEP-CK, and this is pyruvate carboxylase, and this is coenzyme biotin. First, carbon dioxide is activated and transferred to the coenzyme biotin. This step consumes 1 ATP. Then, pyruvate carboxylase transfers carbon dioxide to pyruvate, generating OAA, oxaloacetate. Oxaloacetate cannot cross the mitochondrial membrane, so it is reduced to malate. Malate crosses the membrane and arrives in cytoplasm. In the cytoplasm, malate is reoxidized to OAA. OAA is then decarboxylated to PEP with consumption of 1 GTP. Carboxylation of pyruvate occurs in the mitochondria and is catalyzed by pyruvate carboxylase that requires biotin as a coenzyme. Biotin is attached to the protein through the epsilon amino group of the lysine residue. This step consumes 1 ATP. The product, OAA, is then transported to the cytoplasm through the malate shuttle. Recall, this is one of the shuttles for transport of reducing equivalents between mitochondria and cytosol. In the cytoplasm, malate is reoxidized to OAA by cytoplasmic malate dehydrogenase. For decarboxylation of OAA, oxygen from the keto group is bonded to phosphate group from GTP, while the carboxyl group is removed. This reaction is catalyzed by PEP carboxykinase, or PEPCK. The enzyme fructose 1,6-bisphosphatase removes one phosphate group from fructose 1,6-bisphosphate and convert this compound to fructose 6-phosphate. The enzyme is found in the liver and the kidney. Glucose 6-phosphatase removes the last phosphate group. This enzyme is in the lumen of the ER. Glucose 6-phosphate is transported into the ER, and free glucose is transported back into the cytoplasm, from which it leaves the cell through GLUT2 transporter. Glucose 6-phosphatase is only in the liver and the kidney. Glucose 6-phosphatase is a complex of 4 proteins, 1 catalytic subunit, and 3 transporters. This is the ER membrane, and this is the ER lumen. Glucose 6-phosphate is first transported into the lumen of ER through T1. Phosphate is removed by the catalytic subunit. Phosphate is transported back to the cytoplasm through T2 and free glucose by T3. Mutation in G6PC, the catalytic subunit, cause von Glerke's disease. Gluconeogenesis requires energy. That energy comes from beta oxidation of fatty acids. Although acetyl-CoA from beta oxidation of fatty acids cannot be converted to glucose, it can be converted to ketone bodies as an alternative fuel for cells. Gluconeogenesis is regulated at 3 levels. First, it is regulated by substrate availability, particularly by availability of glucogenic amino acid. Second, it is regulated by allosteric regulation. It occurs at pyruvate carboxylase, which is activated by effector acetyl-CoA. Third is transcriptional regulation. It occurs at PEPCK and G6PC genes by glucagon and cortisol. This slide emphasizes the role of acetyl-CoA in coordinated regulation of pyruvate carboxylase and pyruvate dehydrogenase. Both enzymes are regulated by acetyl-CoA. In the fasting state, oxidation of fatty acids generates a large amount of acetyl-CoA. Accumulating acetyl-CoA activates pyruvate carboxylase and inhibits PDH, whereby it prevents conversion of pyruvate to acetyl-CoA. In the well-fed state, ATP is high in the mitochondria, and it inhibits isocitrate dehydrogenase. Any accumulating acetyl-CoA is condensed with OAA and shuttled into the cytoplasm for fatty acid synthesis. This leads to a low level of acetyl-CoA. Under this condition, pyruvate carboxylase is inactive, and pyruvate is oxidized to acetyl-CoA by PDH. Glucagon and cortisol increase transcription of PEPCK and G6PC genes. Glucagon binds glucagon receptor and activates a signaling pathway which leads to activation of transcription factor CREB. CREB binds the enhancer of PEBCK gene. Cortisol binds the receptor glucocorticoid receptor. Glucocorticoid receptor is a nuclear receptor that acts as a transcription factor. Glucagon signaling and cortisol work together to enhance transcription of both PEBCK and G6PC genes. In fact, transcription of glucogenic genes also require a co-activator, CRTC2. It stands for CREB regulated transcription coactivator 2, or TORC2, shown in this picture. Here is an integration with pharmacology. Metformin, an anti-diabetic drug, activates AMPK. AMPK then phosphorylates TORC2 and sequesters it in the cytoplasm. As a result, transcription of glucogenic genes is inhibited. Gluconeogenesis can be affected by consumption of alcohol. Ethanol is oxidized to acetate by two enzymes, alcohol dehydrogenase and aldehyde dehydrogenase. These two reactions, along with further metabolism of acetate, can generate a large amount of NADH. High NADH favor the formation of the following substance: lactate from pyruvate, malate from OAA, and glycerol 3-phosphate from dihydroacetone phosphate. The effect is to divert important glucogenic substance from entering the glucogenic pathway. Ethanol-mediated increase in NADH causes the intermediates of glucose to be diverted to other alternative pathways. For example, pyruvate to lactate, oxaloacetate to malate, resulting in decreased synthesis of glucose, leading to hypoglycemia. The hexomonophosphate shunt occurs in the cytoplasm of all cells. It serves two major functions. One is to produce NADPH. Second is to provide ribose 5-phosphate for nucleotide synthesis. The first part of HMP shunt begins with glucose 6-phosphate and ends with ribulose 5-phosphate, and it is irreversible. This part involves the important rate-limiting enzyme glucose 6-phosphate dehydrogenase, or G6PD. The second part of the pathway, beginning with ribulose 5-phosphate, represents a series of reversible reactions that produce a pool of sugars for biosynthesis, including ribose 5-phosphate for nucleotide synthesis. Because fructose 6-phosphate and glyceraldehyde 3-phosphate are among the sugars produced, intermediates can feed back into glycolysis. Conversely, pentose can be made from glycolytic intermediates without going through G6PD step. Transketolase, a thymine-requiring enzyme, is important for this conversion. Transketolase is the only thymine-containing enzymes in red blood cells. This information is provided on this slide. NADPH has several important functions. For example, it is required for biosynthesis of fatty acids, cholesterol, and nucleotides. It is required for production of reduced glutathione to protect cells against reactive oxygen species. It is required for generation of bactericidal chemicals to kill bacteria by polymorphonuclear leukocytes, including macrophages and neutrophils. In red blood cells, HMP Shunt is the only pathway that provides NADPH. NADPH is required for production of reduced glutathione in order to remove hydrogen peroxide in red blood cells. Red blood cells are particularly susceptible to G6PD deficiency. When G6PD is defective, oxidation of red blood cell membranes and proteins leads to shape change and rigidity. Heinz bodies appear in red blood cells. Heinz bodies are inclusions of denatured hemoglobin. Eventually, red blood cell will die, leading to hemolytic anemia. Here is clinical correlation. G6PD deficiency is an X-linked disease. It is the most common enzyme deficiency worldwide, with around 200 million people affected. Certain regions in the world have a high prevalence. Common symptoms include neonatal jaundice and acute hemolytic anemia. Several factors can exacerbate the symptoms of G6PD deficiency. One is oxidant drugs that induce hemolytic anemia in G6PD deficiency, known as AAA. It represents antibiotics such as sulfur methoxazole, chloramphenicol, antimalarials such as primaquine, antipyretics such as acetonide. Second, certain food such as fava beans. And third, infection. Inflammatory responses to infection result in the generation of free radicals in macrophages, which can diffuse into the red blood cells. Finally, NADPH is required for generation of bactericidal chemicals to kill bacteria by phagocytes, including macrophages and neutrophils. After bacteria are phagocytosed in the phagolysosome, killing bacteria requires a large quantity of reactive oxygen species in a short period of time, which is known as respiratory burst. Respiratory burst requires two enzymes. First, NADPH oxidase that generates superoxide. Superoxide is converted to hydrogen peroxide, which is then converted to either hypochlorite acid or hydroxyl radical. Hypochlorite acid is the main component of bleach. Conversion from hydrogen peroxide to hypochlorite acid requires the second enzyme, myeloperoxidase. NADPH oxidase catalyzes the synthesis of superoxide. This reaction requires NADPH. Deficiency of NADPH oxidase leads to chronic granulomatous disease, or CGD. Patients have recurrent infection. Myeloperoxidase produces hypochlorous acid. Myeloperoxidase deficiency predisposes patients to immune deficiency. Compared with NADPH oxidase deficiency, this is a milder condition.